Jean-Paul Chataravian, a graduate student in the Blurton-Jones lab, and we recently published a new paper in the Journal of Experimental Medicine titled Engineering an Inhibitor-Resistant Human CSF1R Variant for Microglia Replacement. The Blurton-Jones lab is focused on understanding how AD risk genes impact and affect microglia function, and furthermore, the role microglia play in the onset and progression of Alzheimer's disease. Microglia are the innate immune cell of the brain and highly implicated within Alzheimer's disease. Therefore, they're an exciting target for cell replacement therapies within the CNS. However, transplanted iPSC-derived microglia engraft minimally within the occupied niche, and typically only at the injected sites. To overcome this hurdle, we studied specific inhibitors binding to the CSF1R receptor, whose signaling is highly implicated and necessary for microglia function and viability within the brain, and identified that a glycine residue at the 795th position was a possible target for CRISPR editing. We proposed that altering the glycine residue to a slightly larger alanine would result in a prevention of CSF1R inhibition while still allowing for normal CSF1 signaling to occur. This residue was also identified separately but in parallel by Dr. Chris Bennett at the University of Pennsylvania, with whom we combined our efforts to bring this study to publication. We found that transplanted G795A microglia not only engraft and remain viable under CSF1 inhibition, but also expand and proliferate throughout the mirroring brain, completely engrafting within 2 months of treatment. Furthermore, we found that these engrafted G795A microglia remain after CSF1R inhibition had ceased and return to a homeostatic state within the brain. This pairs nicely with our ongoing efforts engineering human iPSC-derived microglia to deliver amyloid-specific therapeutics within the brain. Together, they offer a new and exciting path forward, not only for cell replacement therapies within the CNS, but a possible avenue to engineer microglia as living drugs within the brain.